Good morning, all of you. So today we are here with a new topic, structure and function of MHC molecules. So I am the presenter, Himani Sethi, lecturer in Geology, AB College, Basudevpur, Odisha. This presentation is prepared for the submission of 6th Guru Dakshita program by DAAVV Indore, HRDC Indore. So we are going to study today about structure and function of MHC molecules. So in this today we are going to learn about the introduction, what are MHC molecules, the classes, how many types of MHC molecules are there, that is MHC1 and MHC2, then your structure and function of MHC1 and MHC2, and at the last we will learn about the comparison and the summary, and at last if the questions are there. So coming to the introduction, so the MHC full form is major histocompatibility complex. Here is a term coming as histocompatibility. If I'm going to show it, the word is histocompatibility. So this histocompatibility, what does it mean? It means that how the tissues of different persons are compatible with each other. Histo, the term has come from the tissue. So this MHC plays an important role in organ where the organ transplant takes place. So just before that, we will quickly go to the what are these MHC molecules. So these are cell surface proteins. So kindly note it down. This is cell surface protein required for adaptive immune response. So this MHC is responsible for adaptive immunity to recognize the foreign molecules. The foreign molecules can be bacteria, can be virus, can any type of pathogens. It also determines histocompatibility. I have already told you what is histocompatibility, and the term histocompatibility has come from this major histocompatibility complex. This is present in all cells, though we will just mention, I would like to mention that MHC1 and MHC2, some are present in the nucleated cells and all other cells are, and other is present in all other cells. So MHC plus antigen, so when an antigen comes and enters inside the cell, the MHC molecule has a function that it will show it to the surface. So MHC plus antigen present on its surface. So these are generally called as alarming molecules. Why they are called as alarming molecules? Because they send a signal to the side by cells, those who are there, that this cell has also get infected. So might be there is a chance that other cells will also get infected. So that's why we call it as alarming molecules. And this MHC are generally proteinaceous in nature. So going to the next slide. So classes of MHC, we have MHC1 and MHC2, and we are going to learn about this two today. But before that, there is other classes are also there. That is one class 3, but generally they are not involved in the antigen presentation. So class 3 proteins comprise a mixture of complement, chemokine and cytokine genes that are not involved in antigen presentation. So today we are not going to look after this. So generally we are going for MHC1 and MHC2. So the genes for MHC protein synthesized are located on chromosome 6 in humans, that is HLA genes. So what are these HLA genes? They are human leukocyte antigen genes. Next, coming to the structure of MHC1. So these MHC1 are encoded by the gene HLA-A, HLA-B and HLA-C. As I have already told you, HLA stands for human leukocyte antigen. So MHC1 plus antigen will be identified by CD8 plus cell. So here there is an important thing that you have to note it down, that CD8 cell and there is presence of CD4 cell. But MHC1 is only recognized by CD8 plus cell. CD4 plus will not, and will not identify this MHC1 molecules. So if going to the structure, so this you can see that the structure is like this. There is one beta microglobulin, and beta microglobulin is not a part, we can say that, but it is attached covalently to the main domain. So it has alpha domain, that is this alpha domain has alpha 1, alpha 2 and alpha 3. This is your transmembrane, this is your transmembrane, and this is your extracellular, and this is your intracellular. This is intracellular and this is extracellular thing. So what we can say that this MHC1 is present in all nucleated cells. I have told you that you have to remember one thing, that MHC1 is present in all only nucleated cells. So class 1 MHC is a glycoprotein, or glycoprotein, we mean to say that glycoprotein means a glycan part is there and a protein part is there. That's why it is called as glycoprotein, and it contains an alpha chain and a non-covalently attached beta microglobulin. So alpha chain has 3 domains, that is alpha 1, alpha 2 and alpha 3. And this beta microglobulin alpha 1 rests upon this beta microglobulin. So peptide binding is between this, that is groove between alpha 1 and alpha 2. So peptide is between, this is alpha 1 and alpha 2. This is a groove, and this groove binding will takes place between alpha 1 and alpha 2. That is peptide binding groove, we can say that. And this MHC molecule, this is present just outside of the cell membrane. Just I have told that this is your intracellular part, this is your extracellular part, and this is your total is the transmembrane. So whatever virus or bacteria has infected the cell, they will process it, and this MHC molecule will take, suppose this is a processed, I'll take some other color, okay, let it be green. So this is a, suppose this is a processed virus or a bacterial part. This it will show it to some other T cells. It can be TC cells, generally it is TC cells. Sorry. So it is TC cell or T cytotoxic cells. So this is all about your structure of MHC. Now coming to the function of MHC. So the main function is binds with the peptide antigen and display them for identification by T cell. This also gives a production of antiviral protein, digestion of foreign materials, and formation of memory cell. Yes, this is very important, that it also helps in the formation of memory cells. So how this memory cells will turn up? So this memory cell, suppose you have been infected with a particular type of disease. So for that sorry, for that particular type of disease, your memory cell has developed already. So if by chance you've been affected with the same disease for the second time, the already immune response has evoked, and it is already there that after what thing they have to do, means the steps has already mentioned by this memory cells. So here with the diagram it is very clear that this is a natural killer cell, again it is a immune cell, and this is a very healthy cell, you can see that, and I'll take a blue one, and this thing is a MHC, this is also a MHC, okay. So this MHC molecules, what they are doing is they are presenting, this is the infected things, it may be a bacteria or it may be a virus. So they are presenting it to the NK cell, which is a natural killer cell, and it will destroy this one. So this will remain healthy. But if the virus is attacked and there is no one to represent here, can you see any MHC molecule? There is no MHC molecules to show it to the natural killer cells, and hence this cell dies. This cell completely dies, but here the cell is healthy. Understood my point? Okay. Next we are going to structure of MHC2. It is very similar with the MHC1. But you can see that there is an extra domain that is beta 1 and beta 2. There beta microglobulin is there, but here it is beta 1 and beta 2. And here the N terminal lies outside and the C terminal lies inside. As we know that it is proteinaceous in nature, so the proteins are having one amino acid. We know that it is having an N terminal and the C terminal. So this is the plasma membrane, this is the extracellular one, this is the intracellular one. So the groove between the peptide binding groove is again between this is alpha 1 and beta 1, but in that case MHC, if you remember, it is alpha 1 and alpha 2. Okay, so beta, but here the groove is between alpha 1 and beta 1. So the MHC2, again there's a difference, is that this is whereas in MHC1 it is encoded by HLA ABC, but here it is HLA DP, PQ and DR. And those MHC were present in all nucleated cells, but these MHCs are present in antigen presenting cells. Generally we say that it is a dendritic, then your dendritic, then macrophages and T cells. Okay, and in that case remember it is CD8, but here it is CD4. Here students, I'll just let you know about just to how to remember generally, because we have a lot of things to remember. So I'll just give one rule, that is 8 rule. Just remember this number 8. So if MHC2, so 2 into 4. Okay, so CD4 will be recognized by MHC2. And in that case MHC1, which one? CD8 rule, so that will be CD2, CD4, okay, CD4, recognized by CD4, and that is by MHC1, 1 into how much is 8? That is 8, so it will be CD1, okay, so one is CD4 and one is CD8, so that will be MHC1 will be recognized by CD8 cell and MHC2 will be recognized by CD4 cell, because if you take the 2, so 2 into 4 8, and if we will take MHC1, 1 into 8 is 8. So this is 8 rule. This is for making easy for remembering. Okay. So next we come as functions of MHC2. It is the same. Here also we can see that with the diagram. It's very clear that this endocytosis, that means inside the cell the virus has came and it has then the processing has taken place over here, and the fusion with Golgi has taken place, and in the Golgi surfaces we can see this MHC molecules. These MHC molecules are there, MHC2, because the structure is showing MHC2 kind of thing, and then it is going to present to the CD cell 4. CD4 means definitely it will be MHC2. This will be MHC2. अभी 8 का rule हमने देखा exactly. तो 4 into so MHC2. So 2 into 4 is 8. So this will be CD4 cell. So to present the functions goes like this. To present processed antigens, I have already said these are the processed antigens which are derived primarily from exogenous source or endogenous source to CD4 T lymphocytes. This is also critical for the initiation of antigen specific immune response. Next, we are going to the compare. Quickly we will compare location of antigen. Here it is endogenous class 1 and this is exogenous. Okay, antigens present in the fluids generally, and here class 1 is antigens from virus or bacterial. Antigen processing is all nucleated cell, but here I have already told you it is APC cell, antigen presenting cell. Here it is clearly mentioned APC and here it is clearly mentioned it is nucleated. And which is the epitope here? I have already told you MHC1 is recognized by CD8 and MHC2 is recognized by CD4. This is T cell cytotoxic cell and this is T cell T helper cell. This is TC and this is TH. Now cytotoxic T cell, this is self cell is making abnormal proteins and must be destroyed. Helper T cells, that a foreign antigen is in the fluid of the body and will release chemical signals to recruit additional immune response to the area. So quickly we have gone through the class 1 and class 2 MHC differences. Now at the last summary, we have learned about the MHC, the classes of MHC, the structure of MHC, the function of MHC, and comparison basis of the mode of the action. That how these two are, one is exogenous and another one is endogenous. So if any question don't feel afraid to ask, I'm there. So